and today we're going to learn more about this thing called RNA you've been writing down in your notes. But let's do a quick little review. Remember, DNA is what your chromosomes are made of, and it's in the nucleus. And DNA has this code that tells an organism everything from what kind of heartbeat to what color eyes if it's a human, to how you digest food. Every cellular process comes from the code, the instructions inside this nucleus, the DNA that makes up your chromosomes. So you may be wondering, how does the message get out to the cell then if it stays in here? Well, it has to send a message. It sends a message through mRNA, and that's what we're about to learn about. So remember, DNA is two-sided. RNA is one-sided, single-sided. So it can actually get out of this nuclear membrane and get out into the cell and tell it what to do. It sends what I often call a work order, what the boss says to do. So DNA has these four bases, A, T, we practice this, C and G, T standing for thymine. But what's different about RNA is it has U instead of T, uracil. It does not have a T. So instead of placing a T, it's replaced with a U in RNA, and it just has a single side. So if I gave you a DNA sequence, you should be able to tell the difference in those two on paper on a test because RNA will have U in it. Okay, so AU on this one, and also CG is how it will message. So how does this happen? The DNA will unwind, and one side will be the template or the pattern to make the RNA, the message, the mRNA, that will leave the nucleus and go out into the cell. So let's practice this just a little bit. We're going to do a few. So set up your sheet of paper. I want your title for this to be RNA Transcription. That's the step we're doing now. RNA transcription. So let's look at just this one. Just this one. So when the DNA unwinds and comes apart, and here's the side we're going to use for a pattern. So remember, it's red in codons. That'll be important a little bit later. But if on DNA, if it's C on this side, it'll be G on this side, right? Now, if the DNA is A, remember, it's going to be U in your RNA. A, U. Okay, so if there's a C, again, we'll have a G. If there's a G, it'll be a C for the RNA side, and the A, again, will be a U. C, G, C, G, T, A, T again, A, and the C is a G. Okay? So that's our first one. Now, as we're setting these up on your page, I want you to leave some space right here for a third column. We'll be writing some words out here in the next lesson when we learn about the next process or next step in this process. So go ahead, if you can get a shot of this one, the second one, please. You pause it right now and copy this down on your notebook paper, please. Okay, we're back. Let's practice this one now. So if there's a T on this side, we'll have an A, T, A, T, A. Okay. Again, we have another T, A, C, G, G, C, G, C. T, A, U, remember, C, G, C, G, C, G, A, U, T, A. Okay. Now, this third one, we can get a good shot of it. Pause the video, copy it down, work it yourself right now. Okay, we're back. Let's see how you did. I hope you paused it and worked it yourself so you would get some practice. So let's see. The T will be an A, U, G. T, A, C, G, U, C, G, 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 A, U, C would be G, G, T is A, U, G, A. How'd you do? Did you get those? All right, one more time. Let's pause it here on the fourth one. Pause that, write that down in your notebook paper, and you work it. Okay, so T, A, U, C, G. C, C, C. U, C, G, C, C, C. A, U, C, C, G, G, A, U. You look funny. U, A. Okay, how'd you do that? All right, so keep up with this. We're going to use this for our next lesson. So keep this paper handy for next time. Thank you.